Hello. Today we'll continue our lecture on the clinical immunological disorders. We'll deal with two subtopics: autoimmune disorders and the transplantation immunology. Autoimmune disorders. It occurs as a result of a breakdown in the regulation of the self-tolerance state, like clonal deletion, clonal energy, or peripheral suppression. So humoral and cell-mediated immunity will work against our own tissues. Tolerance to self-antigen is specific for the inducing epitope and is more readily induced and lasts longer in T cells than in B cells. Suppression of autoreactive T lymphocytes have recently been attributed to the conversions of a CD4 T lymphocytes into T regulatory by an important protein called FOXP3. It's a suppressive protein and as well it's a transcription factor. Tregs produced by a normal thymus are termed natural, while Tregs formed by the differentiation of the naive T cell outside the thymus are called adaptive. We can see here from the sketch that the T regulatory, once increase in number, it will promote cancer. Why it will promote cancer when it's increased in time in number? Because in cancer, an excess of Tregs activity can prevent the immune system from destroying cancer cells. So once it increases in number, it will add to the preventing the immune system, so cancer will prevail. While this compromise the suppressive function of the Treg will reverse the effect, and it is a strategy used to treat cancer. In the normal case, Tregs prevent autoimmunity by its suppressive function. So once we increase or enhance this suppressive function of the Tregs, this will promote more inhibition to the autoimmunity, because in autoimmune disease, a deficiency in Tregs activity can allow other autoimmune cells to attack the body's own tissues. So, first subtopic in autoimmune disorder is self-tolerant state. So, first, clonal deletion. Clonal deletion, when the immature CD4 T cells, which bear the receptors of indigenous molecules, are deleted after contact with self-antigen in the neonatal thymus gland. The immature B cells with a self-reactive receptor likewise are eliminated after a contact with potential self-antigen in the bone marrow, a mechanism that ensures that nothing will go out of the bone marrow unless it is not reactive to the self-receptors. While clonal energy is the loss of T cells and B cell functions after exposure to antigen in the absence of mandatory signal, or following exposure to cells lacking major histocompatibility complex class II molecules. For peripheral suppression can occur if CD8 T cells or macrophage secrete cytokines like TGF-beta, which downregulate the immune system. In this figure, you can see a sketch for the difference between energy and deletion and suppression. You can see that energy can use like inhibitory receptor that do functional unresponsive, make the cell functionally unresponsive, while deletion, it eliminates the cells that can react to the self-antigen by the process of apoptosis, which is activation-induced cell death. While for the suppression, under the effect of growth factor or signals like TGF-beta, we have just said, it blocks the activation of the immune cells. Second subtopic, autoimmune disorder can occur in several ways varying with the order. Like first thing is microbial antigen. Example, streptococci antigen cross-react with human tissue antigen, like the sarcolemmal heart muscles and kidney. So the body recognize that and generate antibodies that may attack the heart muscle and kidney. In other cases, the antimicrobial DNA antibodies react with cells from patients with SLE. Although an autoimmune reaction against the patient's tissue is induced, but still the antigenic stimulus is of exogenous origin. What about hidden antigenic determinants? What's hidden antigenic determinants? Actually, it is it's a kind of engine that, for example, that were unavailable when fatal clonal deletion was occurring and become available following damage by surgery. As well, you can see it as an example in patients with rheumatoid arthritis who form rheumatoid factors, which are mainly IgM antibodies against the Fc region of IgG. So to clarify more on the hidden antigenic, like the case which exposed, you can see here example that physical trauma to one of the eye result in the releasing of sequestered or hidden intracellular protein antigen. Once released in the circulation, it can be recognized by the lymph nodes and activate T cells. In this case, the effector T cell return via bloodstream and attack the antigen in both eyes. So this kind of engine might be in organs that during development have escaped, having been in contact in the reticuloendothelial tissue. So in this case, the embryo or the fetus can be born with this engine inside his eyes. So once there is a surgery or a trauma to the eye, this releases the antigen to the bloodstream and got recognized. So to preserve the patient's the other undamaged eye, it is necessary to remove the damaged eye to prevent further flow of the antigen to the lymph node and to give immunosuppressive agents or drugs to interfere with the ongoing immune response to save the other eye from destruction. So in case of the rheumatoid arthritis, for unknown stimulus, the B cells, unknown stimulus like genetics or hormonal or psychosomatic factors, the B cell is differentiated into plasma cell. These plasma cells secrete very specific antibody IgM, which is actually it's anti-IgG, and this one is termed the rheumatoid factor, RF. This IgM can coagulate with the IgG forming a complex. The RF itself is not specific for rheumatoid arthritis, and it's also seen in other immune disorder. Once the complex is formed, IgM-IgG, it can provoke a complement activation that leading to inflammatory cytokine release. These inflammatory cytokines will damage the synovium. Tissue and other than the synovium may be the site of this as well complex, IgM-IgG complex, as tissues like blood vessels, lungs, heart, and nerves. Absorption of a foreign hapten like quinidine or sulfaziol. Absorption of an endogenous molecule or cell like platelets leads to the formation of an antigenic hapten carrier complex. Antibody is formed and react with the drug on the platelet membrane and this lead to complement activation leading to platelet lysis. From this figure you can see that a carrier like a red blood cells can have an hapten bound on its surface. This hapten will have a multiple epitope site for the antibody recognition. So you can see the binding of this hapten or drug on the surface of an endogenous molecule or a cell, like here the example is red blood cells, can produce sites for the for antibody to behave like an autoantibody, and they will attack the hapten as well as the red blood cells. If the normally occurring suppression by the Treg, because as we initially said that Treg is like a suppressive mechanism for the immune system and it's a mode of regulation for the activity of the T cells, if the normally occurring suppression by Treg of B cell clones that arise with idiotype specificity for self-antigen is lost or diminished, autoantibodies may result. An inordinate switch from T helper 1 to T helper 2 cell activation unique antigenic stimulus may favor antibody formation. This is as the figure we have just mentioned before. Systemic autoimmune disorder. First one is SLE. It's a multi-systemic disease with major lesions like rash, vasculitis, arthritis, and nephritis. Actually, it's a multiple autoreactive antibodies against diverse cellular constituents are formed. For rheumatoid arthritis is a chronic systemic inflammatory disease that is characterized by pannus formation and subcutaneous nodules in the joints. As well, IgG, IgM complexes activate complement, resulting in lesions. For Sjögren syndromes, it's characterized by autoantibodies formed against salivary ducts antigen, lymphocytic infiltration, and immune complex formation in the salivary glands. The clinical features occur primarily in the postmenopausal women, including dryness of the mouth, trachea, bronchi, eyes, nose, vagina, and skin. It may occur secondary to rheumatoid arthritis and SLE. For a polyarthritis, a nodosa is one of several human vasculitis of varying causes, involve hepatitis B antigen antibody complex, which are found in the vessel walls of 30 to 40% of patients. The fourth subtopic of autoimmune disorders is the organ-specific autoimmune disorders. Blood disorders, actually it's anemia, leukopenia, and thrombocytopenia disorder that possess antibodies to react with RBCs, white blood cells, and the platelets respectively. Multiple myeloma. It is a malignant transformation of a single plasma cell clone in which patients secrete Bence-Jones proteins, which is a monoclonal light chains antibody in their urine, and this is a very diagnostic feature. For the central nervous system disorders, the first one is allergic encephalomyelitis. It's a demyelinating disease occur after infection or immunization, and scientists have succeeded to isolate a nanopeptide from the brain that have been implicated in as the antigen. For multiple sclerosis, it's a chronically relapsing disease of unknown etiology that is characterized by mononuclear cell infiltrates and the demyelinating lesions in the white matter of central nervous system. A decrease in suppressor cell function and elevated titers in viruses appear in CSF, suggesting a virus etiology. For myasthenia gravis, it's a thymic hyperplasia, muscle weakness, and characterized by muscle weakness and fatigue. Actually, it's a defect in neuromuscular transmission is indicated by the presence of anti-acetylcholine receptor antibody. So this leads to the inability to transmit the acetylcholine-induced signal to muscle fibers, results and causes these clinical sign. For the third type of organ-specific autoimmune disorder, the endocrine disorder, its first one is a chronic thyroiditis, Hashimoto disease, or hypothyroidism. It's a property of genetic basis that affect mainly women, characterized by autoantibody and cell-mediated immunity to thyroglobulin or thyroid peroxidase, and leading to the progressive destruction of thyroid gland. ADCC may be responsible for the tissue damage. The other way around disease is graft disease, hyperthyroidism, leading to diffuse goiter and thyrotoxicosis. T and B cells infiltrate thyroid gland and leading to the formation of autoantibodies to the TSH receptor. This complex will lead to competition between the autoantibodies with the TSH and induced uncontrolled TSH-like activity. The third one is diabetes mellitus type 1. It is the destruction of the insulin producing cells in the pancreas, beta cells. Either humoral or cell mediated anti-islet cell activity can be operative, and there is no evidence of an autoimmune disease for the type 2 diabetes. For ulcerative colitis, it is a chronic inflammatory lesions confined to rectum and colon and accompanied by infiltration of monocytes, lymphocytes, and plasma cells. Patients' lymphocytes exert cytotoxic. Scientists have found that the patient's lymphocyte exerts cytotoxicity against colonic epithelial cells when they are growing in culture, and they may have antibodies that cross-react with E. coli, but the disease of unknown etiology. For Crohn's disease, it's a chronic inflammation disease that involves T and B cells, macrophage and neutrophils. This disease usually occurs in submucosal area of terminal ileum. It has often been suspected but has not been established as being of microbial etiology. The second topic of our lecture today is transplantation immunology. This is governed mainly by the allogenic differences in HLAs between donor and recipient. HLAs have two major functions: to bind and present processed foreign antigenic peptides to T cells, thus initiating the immune response, and as well to identify and distinguish the MHC membrane antigen on a transplanted donor organ from those of the recipients. There are two classes exhibit enormous polymorphism since multiple allele exist at each locus, and we have discussed that in previous lectures. For class 1 antigen, it's found actually on all nucleated cells. 3 gene loci control that, A and B and C, and these antigen can be identified serologically with anti-HLA antibodies. And the main function of this class is to present foreign antigenic peptides to CD8 cells, while class 2 are found on immunologic effector cells like macrophage, dendritic cells, B cells, activated epithelial cells, and the antigen is controlled by 3 other genes: DP, DQ, and DR, and they function to present antigenic peptides to CD4 cells. So it is important to do matching the donor and recipient at the HLA locus. This improves graft acceptance. Both donor and recipients are typed for HLA profile, ABO and RH antigen with their respective antisera. The donor must be tested for pre-existing anti-HLA antibodies and CMI, since sensitization to HLA antigens can occur as a result of prior blood transfusions, pregnancy, and other organ grafts. The degree of genetic disparity between donor and recipients governs the vigor and the speed of rejection of the graft. TC cells and macrophage mediate most rejections. For the acute rejection is characterized by swelling, tenderness over the allograft. It is initiated by the HLA antigen on the allograft, stimulating recipient T helper cells, leading to injury to the renal vasculature by TC cells with ischemia of the renal parenchyma, while a second allograft from the same donor as the initial allograft is rejected is more quickly than the initial allograft due to, of course, the memory response of the immune system. For the chronic rejection, it is episodic bouts of rejection occurring months to years after transplantation. And of course, both CMI and HI are active, resulting in interstitial fibrosis, vascular occlusions, and loss of function. For the hyperacute rejections occur within minutes, never takes time because of the pre-existing already sensitivity and rejection occur within minutes. So graft versus host reactions occur when the immunocompetent tissues like bone, marrow, thymus, spleen, organ harboring passenger leukocytes are allografted, like bone marrow transplantation. If they recognize the recipient as a foreign, CMI damage results. If recipient is immunocompetent, a horses versus graft reaction doesn't take place. The immunosuppression. These immunosuppression lead to prolonged graft acceptance. However, it makes the individual susceptible to infection. Consequently, appropriate killed but not live vaccines should be administered before transplantation. Major organisms that cause infections are CMV, Candida, Epstein-Barr virus, Aspergillus, and respiratory syncytial virus. Scientists have seen that CMV present in more than 50% of the donors, while Candida present in more than 90% of the donors. Epstein-Barr virus present in more than 90% of the donors as well, and Aspergillus as well. Respiratory syncytial virus present as well. For the immunosuppressive agents in the transplantation immunology, a lot of drugs have been used as an agent for immunosuppression, including cyclosporine A, azathioprine, and corticosteroids. Thank you.